It's that time again. This is That's Mother Science. How did life begin? It would seem that the complex recursive relationship between DNA, RNA, and proteins presents such a level of complexity that it's quite improbable that these mechanisms would arise spontaneously from a purely prebiotic environment. In the 1960s, it was proposed by these guys that perhaps primitive life was based on mRNA, rRNA, and tRNA. They proposed that solely RNA could be used to carry information, catalyze reactions, and specifically for RNA's own replication. Sounds great, but wait a minute. We are scientists. Intuition is fine, but we need experimental evidence to strongly corroborate hypotheses, especially ones that relate to the origin of life. Since you asked, in 1986, Gilbert showed that RNA can catalyze chemical reactions. More specifically, in the 90s and 2000s, several studies showed that ligase ribozymes could replicate their own code. These instances basically relied on longer nucleotide sequences. The problem is, the longer the nucleotide sequence, the Lego blocks you build from, it doesn't exactly add much support for the notion that the mechanism is viable in, say, a random schlop of prebiotic material. I want to remind you this information is motivated by the introduction in the paper that we're discussing. Without them, my grand comprehension of organic chemistry would have brought you. It's also been shown that RNA polymerase ribozymes could synthesize RNA from template strands. These approaches utilize single nucleotide bases. This does offer support for the prebiotic schlop dynamic because it doesn't depend on giant complex streams of nucleotides conveniently existing in relevant concentrations in close proximity to Mr. Ribozyme. They had demonstrated that linear strands of RNA could be replicated by ribozymes. We'll just pretend that my hand is the ribozyme, and he goes along, chunking along, one nucleotide at a time until, boom, 2 strands. Dumbest thing. But there was one looming elephant of a problem. Let's just imagine for a moment we have a more complex RNA sequence and structure. Let's say like this. This is Mr. Squiggle. By the same mechanism, once again, let's pretend my hand is the RNA machinery in question. Chugging along. Ooh, you're not supposed to see those. Those are prebiotic schlop. Chugging along. Oh, hold on. I can't. My hand's. These higher level structures are necessary for functional machinery using RNA. Using the catalytic domain from the ZRPR, citation, or Z-core, which the researchers deduced would work with nucleotide groups as short as 3 when they incubated in the eutectic phase of water ice. Using Z-core variants, specifically 1.5 times 10 to the 15th of them, they're really small. They put all of their faith in RN Jesus, and they power-leveled their pokey ribozyme. Seriously though, after 7 rounds of in vitro evolution, a fourth of the lot had evolved into 0 core. There were multiple iterations in this study, and this is best articulated by the figure from the paper. Ultimately, culminating into the T5 plus 1 ribosome is a heterodimer, which means it's made of 2 parts, consisting of the catalytic unit T5 and the non-catalytic RNA cofactor type 1. This represents the first instance in which a synergistic cofactor to a ribozyme has been produced spontaneously and they possess a broad capacity to polymerize triplet nucleotides. Now for the reality. Although this progress is pretty important, it remains that the T5+1 ribozyme complex lacks the efficiency to synthesize RNAs as long as itself. Self-replication is the ultimate goal. It was shown that after splitting the catalytic component, T5, into separate segments, it could synthesize combined segment groups that spontaneously yielded new catalytically active ribozymes. This process was afforded nucleotide sequences 8 nucleotides long to perform the task. This is on par with the previous instances. However, in synthesis of other RNAs, it was shown that an accuracy of 97% was attained using pools of just 3 nucleotide groups. And this study produced complex folded structures from RNA where only linear segments had previously been reproduced. That's mother science.